So great turnout today. I think this is obviously an interesting topic for lots of people, which is terrific. So it's my pleasure to introduce Ollie Payne, who is a Sir Henry Wellcome and Wellcome Trust funded fellow from King's College London, who's going to speak on the subject of polygenic risk scores and introduce, I think, software called GenoPred, which is really nice. I've seen Ollie give a talk on a similar subject before, and it was a real pleasure. And the software is really terrific. So, without further ado, I'm going to hand over to Ollie to give his presentation. But I hope you enjoy this even half as much as I enjoyed seeing his talk earlier. Thanks, Ollie, and over to you.
Wonderful. Thank you very much, Andrew. It's a pleasure to be presenting here today. Thank you for joining. As Andrew said, I'm going to be talking about polygenic scoring, specifically looking at polygenic scoring methods that leverage GWAS data from multiple populations when calculating that polygenic score, and then how that kind of sits within our GenoPred pipeline, which I'll introduce in a moment. So starting at the beginning, genetic variation plays an important role in determining a range of health-related outcomes, including risk of disease, as well as response to medication. And to try and understand the genetic basis of these outcomes, we've been carrying out a lot of genome-wide association studies, or GWAS, which scan the genome for common genetic variants associated with our trait of interest. And there's been a huge drive on the GWAS front over the last 2 decades. We now have very large GWAS with millions of participants included for a broad range of clinically meaningful kind of phenotypes or traits. And we can now use those GWAS summary statistics for a range of downstream analyses. And one of those applications of GWAS is called polygenic scoring. So a polygenic score summarizes an individual's genetic risk or propensity for a given outcome by aggregating the genetic effects across the genome. And it kind of weights the effect of genetic variants across the genome using effect sizes from these genome-wide association studies. So they can be considered a kind of GWAS effect size weighted sum of the risk variance an individual carries. And these polygenic scores have been incredibly useful for a range of purposes. They've been used extensively in research to answer all sorts of questions, such as, do 2 traits overlap genetically? What is the genetic architecture of the trait we're looking at in terms of heritability and polygenicity, and many other kind of research questions as well. But these polygenic scores are also emerging as a useful tool for informing personalized medicine. And I'll give a few examples of that now. So one of the things that polygenic scores can be used for is predicting risk of diseases. And there's, due to these very large GWAS being performed, there's now polygenic scores able to provide kind of clinically useful prediction for a range of common traits. And this was nicely illustrated by this paper by Emmett Kira et al., but there's many other studies illustrating this as well. But they show that these polygenic scores can be used to identify individuals in the general population at substantially elevated risk for, I think, 8 diseases. With this figure on the right, showing the example of coronary artery disease, where it shows that the top 8% of individuals in the coronary artery disease polygenic score have a 3-fold increase in their risk of coronary artery disease compared to the population prevalence. So that's quite a large proportion of individuals at a significantly elevated risk, just identified using polygenic scores. And they argue that this ability to kind of stratify individuals by risk is equivalent to that of monogenic mutations that are already currently used in many clinical settings, and therefore polygenic scores could be used as well. So in addition to calculating or predicting an individual's risk for a given outcome, there's also potential to predict other clinically useful outcomes, such as response to medication. And this is an example of this, again, looking at coronary artery disease, where they show that individuals with a high genetic risk for coronary artery disease not only have a higher likelihood of developing kind of disease events like heart attacks, but they also show a larger risk reduction when given statins compared to a placebo drug. And this really nicely illustrates that polygenic scores can be used for what's called both predictive enrichment, but also prognostic enrichment, whereby you're able to predict the likelihood of a disease event, but also the effect size of the medication. So this is useful for improving the efficiency of clinical trials, but it also highlights their utility for predicting kind of groups of patients that might respond best to a given medication. And so polygenic scores can predict a range of outcomes. That's great. One of the concerns has been how we actually integrate that into our clinical settings alongside other information that's already used. And this is a really nice study, again, showing this kind of clinical utility and clinical acceptance of polygenic scores by doing a study where they integrate polygenic scores alongside existing clinical variables when predicting cardiovascular disease risk. And they show that it improves prediction over and above the currently used kind of clinical tools, but also it fit well within that kind of primary care setting. So again, this is quite encouraging. It shows that these polygenic scores are about to kind of emerge potentially, or studies evaluating them in clinical settings are going to start emerging at least, which is great news. However, one of the kind of major limitations of polygenic scores is that their accuracy varies across different populations. So as an example, if you do a GWAS in a European ancestry population, the polygenic score derived from that GWAS will be most accurate in European target samples, but less accurate in non-European target samples. And this is nicely shown in this plot by Alicia Martin. There's several other studies showing the same thing and really nicely documenting this kind of issue. But essentially, this is a big problem because this means that if polygenic scores are implemented within a clinical setting today, they will potentially exacerbate existing health disparities across ancestral populations, as they will benefit individuals within the European population more than other populations. And this is partly due to the high overrepresentation of European ancestry individuals in our GWAS studies. And yeah, that means that, yeah, it's currently going to be most accurate within Europeans. So what are the solutions? Well, one very important thing to do is ensure we are capturing kind of broad ancestral diversity within our GWAS studies. This is, very important. It's important not just for polygenic scoring, it's important for other goals of kind of GWAS and statistical genetics. But yeah, so this is something that's ongoing. There's already brilliant efforts around the world collecting this data. So hopefully we'll see in the next few years kind of even better representation. The other kind of potential solution to this issue of polygenic scores kind of not transferring well to other populations is to try and develop methodology that specifically kind of focuses on addressing this issue by leveraging the data we do have available in different ancestries to kind of maximize that cross-population transferability or portability, as it's often referred to. And it's this second point regarding the methodology that I'm going to talk about today. So over the last few years, we've been looking at lots of research questions relating to polygenic scoring in terms of how they're calculated, how do we adjust for ancestry, how do we kind of put them on a scale that's interpretable. And we've kind of carried forward our findings from that research and put them in a easy-to-use software pipeline called GenoPred. So it's a pipeline that implements a range of the leading methodology for polygenic scoring. It's very easy to use. Once you've kind of specified your target data and your GWAS data, you can run the whole pipeline in a single line of code. It's very robust. It's been tested extensively. It's also highly scalable to very large data sets, but it can also be applied to a single individual's target genetic data as well, if that's what you want to do. Something I like to highlight about it is that it's reference standardized, which means the polygenic score SNP weights, the kind of genetic effects being used in the polygenic score are reference standardized, meaning they're not dependent on the target sample in question. And this is important when using polygenic scores for prediction modeling, as you need to ensure your predictors, your polygenic scores, can be reliably generated in external data sets. And also, I'm pleased to say that, we've made it work for target populations and GWAS data from a range of populations. It can work for any population for which you have sufficient LD reference data, so reference genetic data for estimating linkage disequilibrium. By default, it uses a combination of the 1,000 Genomes and the Human Genome Diversity Project, which gives kind of decent representation. There's a lot of improvement to be made, particularly in African populations, but as a starting point, it means you can already kind of analyze data from around the world fairly easily. But one of the main limitations of it currently is that it, although it implements a range of leading polygenic scoring methods, they are all what are referred to as single source methods, which means they only consider one GWAS when generating the genetic effects used in polygenic scores. But this is a bit of a kind of limitation now, because often we have multiple GWAS from different populations available for a single trait. And currently these single source methods do not leverage that data. So I'm now going to introduce kind of the alternative approaches. First, I'll just give a run through of this single source, these single source methods to then lead into the multi-source methods. So we have our GWAS summary statistics, where we have effect sizes for each variant. We can then put those through a polygenic scoring method like pure SCS, and that will then give you a set of SNP effects, so effect sizes for each variant that can be used for polygenic scoring. These methods often give you multiple sets of SNP weights based on a range of kind of hyperparameters used to adjust those effect sizes. For example, they could be different shrinkage priors. And then the user has to do what's referred to as tuning, whereby they identify which of these sets of SNP effects provide the optimal prediction. And this can be done using either individual level data, where you have an independent sample with both genotype and phenotype data, where you can then test all of these polygenic scores and select the one that provides the best prediction. But, that's often impractical or impossible due to a lack of data. So there's also approaches called that use GWAS summary statistics alone, and I kind of refer to these as SumStat tune. They're also referred to as kind of auto models or pseudo validation models. But essentially, these models are very useful, and I'll kind of talk about these two tuning approaches later in the talk. And then once you've done that tuning, you then have a single set of SNP effects that you can use to calculate polygenic scores in your target sample. So an alternative approach which leverages GWAS from two population-specific GWAS from two populations is to apply a single source method to each GWAS separately to calculate population-specific polygenic scores, and then you can combine them to kind of take advantage of having both those polygenic scores. And you combine them just by fitting them in a single linear model. And that's kind of illustrated here. You depending on the target sample, the ancestry of that target sample, the weights of the population-specific polygenic scores will need to be adjusted. You might imagine a European target would make kind of give a higher weight to a European polygenic score, whereas if you're predicting into an East Asian ancestry target sample, then you might want to upweight the East Asian polygenic score instead. So this is one approach. I refer to these as independently optimized because the SNP weights that are used in the polygenic scoring are based on one GWAS at a time, and the GWAS are not analyzed together. In contrast, there's kind of some other methods which specifically aim to jointly analyze GWAS from multiple populations when deriving those polygenic score effect sizes. And an example of this is pure SCSX. There's other methods as well, which I'll introduce in a moment. But essentially, they argue that this joint optimization and joint analysis of the GWAS improves the predictive utility of those polygenic scores. And similarly to the independently optimized multi-source methods, it produces population-specific polygenic scores, which then need to be linearly combined to give you a final polygenic score for estimating the target sample. One thing I forgot to mention is these independently optimized multi-source methods are denoted by writing the name of the polygenic scoring method you're using, for example, pure SES, but then appending multi to the end, indicating that you've used pure SES polygenic scores from multiple GWAS. So, with all of that in mind, the aims of our study were to try and understand which polygenic scoring methods work best in GWAS from different populations. They've been extensively analyzed in European populations, including in a previous study of ours, but I don't think there's been enough research looking at the accuracy of these methods when using African and East Asian GWAS summary statistics. Then also, we need to compare these different multi-source approaches. Are the jointly optimized methods better than the independently optimized methods, given different methodological kind of strengths of the two approaches? And also, what summary statistic only tuning approaches are available when you are in this multi-source setting. And I'll talk about that a little bit in a moment. And then finally, once we've done all of these kind of benchmarking, we will then implement the leading methodology within our GenoPred pipeline to broaden their accessibility and kind of make more equitable polygenic scores something that people can easily calculate. So the design of our study was to use GWAS summary statistics from three populations, including African, East Asian, and European. The African GWAS summary statistics came from the Uganda Genome Resource. The East Asian ancestry GWAS came from Biobank Japan, and the European ancestry GWAS came from a European subset of the UK Biobank. We then applied a range of polygenic scoring methods. So we have the single source methods here. Then in the multi-source side of things, we have the kind of the independently optimized multi-source methods, whereby we're just using the single source methods, but fitting them together. And then we have the jointly optimized multi-source methods like PRSCSX, X-Wing, and TLPRS. Then we have the tuning approaches that we compare, where we use individual level data to tune the polygenic scores that come from these methods, or GWAS summary statistics alone. And we finally evaluate those polygenic scores in target data from UK Biobank, split into African, East Asian, and European ancestry. So one thing I've mentioned is this issue of tuning your polygenic scores. And one of the main limitations of the independently optimized multi-source methods is that there's currently no approach for tuning the linear combination of those population-specific scores without individual level data. And this is kind of a limitation. So this was one of the things we wanted to address, and we did so by extending some methodology called Leopard to work with the output of single source polygenic scoring methods. So this is a kind of novel extension of an existing method that we evaluated as a method for estimating the optimal linear combination of population-specific polygenic scores. So we can come back to this if anyone's interested, but I wanted to highlight this as an area of kind of novelty, and the results I'll show later. Okay, so on to the results. So what I'm showing here is the average correlation between the polygenic scores and our outcome phenotype or trait across 10 different traits. I forgot to mention, the 10 traits we selected were selected based on their heritability and polygenicity to represent a range of genetic architectures which could influence the performance of the methods. And what I'm showing here is the performance of those polygenic scores when using, in the top box, using in an African target sample, in the lower box, an East Asian target sample. And I'm first of all using target matched GWAS summary statistics to calculate those polygenic scores. So here we have polygenic scores derived using African GWAS, predicting in an African target, and here we have the East Asian GWAS predicting into an East Asian target. And the colors of the points indicate whether or not the polygenic score was tuned using individual level data or using summary statistics alone. So what do we see? First of all, LDPRED2 performed well in both African, when using both African and East Asian GWAS summary statistics, when using individual level data to tune the polygenic scores. Interestingly, the PT plus clump, the p-value thresholding and clumping approach, which is a kind of less sophisticated polygenic scoring method, performs relatively well when using the African GWAS summary statistics, in contrast to when using the East Asian GWAS summary statistics. And this is kind of also in contrast to when using European GWAS. So this is something to think about. Why is this method doing better when using the African GWAS summary statistics? When you're using African GWAS, in this study at least, it appears that methods that tune the polygenic scores using summary statistics perform worse than they do when using East Asian GWAS summary statistics. And again, this indicates there's something specific to these GWAS that we're using and the methodology we're using that is kind of reducing their performance. Something important to note is, so we have the different methods on the x-axis. In this final column, it says all. This is evaluating a model where we considered polygenic scores from any method. So this really indicates, what was the best performance for each trait looking across all methods combined. And we see there's an increase, a greater increase in the African context provided by this all model, which indicates no single method consistently performed best across traits. Whereas in the East Asian target, it performs very similarly to the best method, which was LDPRED2, indicating LDPRED2 performed best or very close to the best method across traits. So now I've just extended this figure to show the performance of polygenic scores when using European GWAS summary statistics. And what we see here is kind of a similar pattern to what we saw when using the East Asian GWAS summary statistics down here. But the performance of S-Bayes RC stands out in particular, now performing better than every other method. And that's in spite of the fact this method doesn't require any individual level data to tune the polygenic score. So this is quite a remarkable performance for S-Bayes RC. And yeah, so this is a newer method, so it was nice to see that it's performing well, and this is now in GenoPred, so all the users can use it easily. Okay, so now in this final section, I'm showing the results when generating polygenic scores using GWAS from both populations. So both the target matched GWAS and the European GWAS. And the first thing we can see is that on kind of looking across methods, the performance of the polygenic scores that leverage GWAS from both populations are better than the polygenic scores derived using either the target matched or European GWAS alone. And this is just, it just emphasizes the point that people should be using all of the GWAS data available when calculating these polygenic scores. So now just zooming into these kind of multi-source results, specifically, what do we see? Well, the performance is quite similar across methods, and this is particularly true in the East Asian target sample. And this is, something to bear in mind is that often the predictive utility is quite similar across methods, and the computational efficiency of the methods may be kind of more important. We'll talk about that in the next few slides. But overall, LDPRED2 again performs best. So this is LDPRED2 multi now. So it's a polygenic score derived using GWAS from European and the target matched GWAS. So LDPRED2 performs very well when you have individual level data to tune the polygenic scores. But if you don't have that individual level data, then S-Bayes RC multi seems to perform very well. And again, there's kind of no method that consistently performs best in the African target sample, indicating that looking across multiple methods may actually be worthwhile if you want to maximize prediction. Now, one thing I want to highlight is that I said at the beginning, these jointly optimized multi-source methods, which kind of consider both GWAS simultaneously, actually seem to not perform better than the independently optimized multi-source methods. And this is in contrast to some previous research, but in line with other research. And essentially, I think it's due to the fact that although these methods do have some advantages by leveraging GWAS from multiple populations together, they are all based fundamentally on the underlying method of pure SCS. And there's certain methodological advances that have been made for other methods that pure SCS may not be taking advantage of yet. So I think currently the other methods such as LDPRED 2 and SBase-RC might have kind of methodological advantages over these jointly optimized methods that currently mean they still perform them. This will likely change. I'm sure there'll be methods coming out in the short term, which kind of take advantage of all these different methodological considerations together. But as it stands, they do not perform better. And in the case of the African target sample, they actually perform a little bit worse. So, one of the things I've kind of mentioned is this using summary statistics alone to tune the polygenic scores, and I highlighted that these independently optimized multi-source methods don't have a method to tune the linear combination of the population-specific scores available. So we developed one, and I mentioned this. It combines a method called LEOPARD with another polygenic scoring method called QuickPRS. And essentially what we show is that the weights of the population-specific scores are pretty consistent across polygenic scoring methods, which makes sense because their performance is also quite similar in absolute terms. And we show that combining QuickPRS with LEOPARD in this plot, we see that there's really very little decrease in prediction accuracy when using this summary statistic tuning approach compared to an individual level tuning approach. So this provides a novel method to derive a polygenic score leveraging multiple GWAS without any individual level tuning data at all. That performs very well, and it's also very fast, which I'll show on the next slide. So I've talked about computational resources. Here's a table showing the time and memory required for each method per GWAS, and things to highlight. So pure SCS and also the jointly optimized methods pure SCSX and X-Wing, which use pure SCS under the hood, are all quite slow. They're all a lot slower than the other methods, and that's something to bear in mind. Given their performance is not seeming to be any better, you may want to use another method. In contrast, our kind of methodological extension of LEOPARD plus QuickPRS to estimate that optimal linear combination of population-specific scores is very quick. It takes about 15 minutes per GWAS, making it very, applicable. And just to highlight some of the time differences between these methods, for example, if you use LDPRED 2 multi, it would take about 45 minutes, whereas if you use pure SCSX or X-Wing, it will take about 14 hours, and that's on 10 cores. So this is, very big difference in computational resources, which may actually inhibit their use in some circumstances. And within the setting of using summary statistics alone to tune the results, then you see even bigger differences. So SBase-RC multi plus the LEOPARD QuickPRS method to estimate that linear combination, about 1 hour and 15 minutes. Pure SCSX, in contrast, is about just under 3 hours, whereas X-Wing in this setting is about 70 hours. So again, our approach of using independently optimized multi-source methods with a LEOPARD plus QuickPRS sum-stat tuning approach is the most efficient and provides optimal prediction accuracy as well. So in summary, we using GWAS from multiple populations improves prediction. That's been shown in previous studies, but we're just driving that point home. Second, we showed that actually currently using independently optimized methods performs just as well as, if not better than, the jointly optimized multi-source methods, but these independently optimized methods are much faster. So that's worth bearing in mind. And our new extension of Leopard to use quick PRS to estimate the optimal linear combination of population-specific scores provides accurate and efficient approach for doing so. So that's nice. So the recommendations based on all of this is that if you have individual level tuning data, then we'd recommend using a combination of LD-PRED2 and SBase-RC, as these methods consistently perform very well. And then you can, look across those methods to see which one's best. If you don't have individual level data, then I would suggest using SBase-RC with the Leopard plus QuickPRS sum-stat tuning approach. So there are some limitations of this research, and I think they should be considered going forward. The main one really in my mind is that we're using very broad ancestry definitions currently. Particularly the African population is very diverse, and it's currently being very very roughly kind of represented by the reference data we have. And this may explain some of the differences we see between the methods when using African GWAS summary statistics. So as the reference data improves, it may be that some of the methods that perform better in the European and East Asian target samples, like SBase-RC, may actually start to perform very well in the African samples as well. So that's one thing to bear in mind. It's not something we can address right now, as the reference data is currently quite poor for African populations. So, but I know a lot is being developed, so this could be something people look at in the future. Another thing to consider is that we've only used HapMap3 variants throughout our analyses. So this is a subset of variants of about 1.1 million genetic variants. They've been used by a lot of polygenic scoring methods in the past because it provides a kind of computationally tractable number of SNPs to analyze. But there are faster methods now, and they can cope with denser coverage of the genome. And actually, some of the methods, particularly those that use functional genomic annotations like SBase-RC, as well as MegaPRS and QuickPRS, it's been shown that these methods kind of gain even better relative performance over other methods when you use a denser SNP set in your polygenic score. So I don't think this will affect the conclusions of this study, but it could actually make them even better than the other methods if we had used a denser SNP set. And finally, in this study, we're talking primarily about a scenario where you have two population-specific GWAS available and you want to analyze them together. But there's also many multi-ancestry meta-analysis GWAS where there's kind of mixtures of ancestries within a single set of summary statistics. We don't look at that in this study. The current advice in the literature is to use LD data from the population that represents the majority of your GWAS, or if it is very mixed, you could try and derive an LD reference that matches the proportions of the ancestries in your GWAS. But that's something we don't talk about here because it's kind of a different issue. Some of these methods, these methods aren't really applicable there. So this study is about to be submitted, and I'm currently integrating, all of these methods have already integrated into the GenoPred pipeline. I haven't released it into the kind of main branch yet with documentation, but that should be coming out very soon. So we've implemented all of these multi-source methods and provided reference data that's automatically downloaded to make it easy for people to implement them. During that process, we also added in a couple new methods such as our Leopard plus QuickPRS tuning approach, SBase RC and QuickPRS, which are two newer polygenic scoring methods. We've also included a continuous ancestry adjustment when calculating polygenic scores in the target sample. This is again trying to make the polygenic scores kind of more interpretable across different ancestral contexts. And something we're working on right now is building in functionality for calculating stratified polygenic scores. For example, pathway-specific polygenic scores, which are kind of picking up in popularity. And I think they have a lot of very useful they provide a lot of interesting opportunities for research as well. So I'll end it there. I would like to thank everyone that's kind of supported me during this study. Received very useful comments throughout from Michelle Camp and Florian Privé. I'd like to thank Catherine Lewis, who supported me when we were developing GenoPred initially, and her team, who are still providing useful feedback as it develops, as well as thanking my fellowship support team, mainly Amar Al-Chalabi, who's my main sponsor at King's College London, but also my research sponsors who have contributed over the years. And massive thank you to King's College London and the Wellcome Trust. Yeah, thank you for your attention. Happy to answer any questions.
Thank you, Ollie. Terrific talk. Everything we'd hoped for, and yeah, wonderfully clearly presented. We've got a couple of questions already, I think.
I was kind of wondering if you've explored these performances of polygenic scores in like other populations that tend to have a lot of kind of diverse genetic or like ancestry, such as the Latin American populations or any indigenous ancestries.
Yeah, that's a really great question and something that is very much in my mind. So in this study, we haven't looked at kind of the admixed American, Central American populations. I think this is something we definitely need to start incorporating. There's different approaches to it, and I would be really keen to kind of go into a few of them in the next kind of couple of years. One is this kind of approach of using local ancestry to pull apart these admixed individuals to provide population-specific GWAS that could then be used in the same way I've used these methods here to calculate population-specific scores. And I think that's probably the best way to go about it, to be honest, right now is to try and separate these different regions of the genome into specific populations, because that will allow you to model the LD appropriately. But then there's the other issue of how do you actually calculate the polygenic score in a target sample that is admixed? And this presents a kind of whole other set of issues, which is currently we use one set of SNP weights to calculate polygenic scores for everyone or within a given population, but perhaps we should be considering the local ancestry again there to use weights that are appropriate to each locus within that target person. So, you could upweight the European polygenic score within European segments, but upweight you see how it goes. And so I think those are two things that are very, very kind of interesting. There's been a couple papers, I think, looking at this kind of second part of estimating polygenic scores within admixed targets, but I think we should see a lot more of that soon.
Yeah, thank you. Yeah, it's a very complicated problem, and I know there's like more efforts more recently, especially for Latin American GWAS and kind of collecting that data in like PGC and things like that. So I was curious if you kind of explored, because it's very interesting that including the European and like target kind of um, or matching GWAS both tend to improve. And I know, like I'm in Canada, so we have an indigenous population that doesn't quite fit with even the like American indigenous population genetics wise. So I was kind of curious if there are different combinations of these GWAS data that, even if they're not specifically for our Canadian indigenous people, if there would be like improved performance if we use kind of a variety of other GWAS. It's very interesting.
Yeah. No, I think that sounds like a very promising approach. Um, yeah, got to piece together the most relevant information for each person, not make broad strokes.
Thank you.
Great. So I've got a question just sent direct to me, and it's asking you to speculate on what the kind of long-term long-term future of polygenic risk profiling and how things will develop, and whether it will expand to include other forms of omic data, which obviously come with their own challenges.
Yeah, I mean, it's a very interesting field. I think it's got a lot of potential. There's a lot of research showing the predictive utility of polygenic scores. The issue they face issues, but I think the more we look, the we see these issues are resolvable. Um, there's been some really interesting work recently looking at overlap of causal effects across populations, and actually it seems like they are fairly highly correlated. We just need to get to the causal variance, and so these kinds of things are, I would say, making me quite optimistic about their utility. Um, in terms of other omics scores, again, I think they're very interesting. They are emerging, as far as I'm aware. There's some really interesting stuff going on with kind of expression scores and methylation scores. I want to see, there probably already is research on it as well. I'm just falling behind, everything moves so fast. But I want to see things about how these scores change over time for a person, for an individual, how they change in different contexts, cell types. Do they alter when you give can you stratify them to predict treatment response by kind of highlighting pathways these drugs target? I think there's loads of potential. Um, there's already studies showing the predictive utility of omics scores, but I think there's so much more we can do. Um, I think, yeah, this is something I'm keen to look at more as well.
Great, good. Good. I have a question. I have a question of my own, but I wonder if there's anybody else in the call wants to. It's not about polygenic risk scores. It's about software development. So, is there anybody else on the call who'd like to put their hand up and ask a question? Well, I'll go ahead, and then hopefully somebody else will have something else. So, I see lots of positive feedback for your software. GenoPred looks like a pleasure to use. It looks very, very well developed and also extremely well presented. And that I think it comes across as something that has been, developed very professionally. And I just wondered if you'd say anything more about that. About you've obviously put a lot of effort into making this something like something a bit closer to what you might pay for. It looks of the quality of something that you might pay a subscription for, or you might actually pay for. I guess I could ask you whether that's likely to be the case in future. But just what led you down that path of such rigor in the presentation and development of the package?
Yeah, well, thank you very much, Andrew. I appreciate the kind feedback. I This just kind of came from the work, and it's something that I really enjoyed doing. I've always been interested in kind of analysis of data and methodology and trying to understand what works well when. I think this is probably partly because we work in psychiatry. It's so hard to know whether it's working or whether the data is just not good enough. And this is something always kind of bugged me. So then I took a step back and started trying to benchmark everything. And then that process of benchmarking everything within a very constrained environment naturally led to the development of workflows that other people could use. So this is the kind of evolution of it. I really enjoy it. Like, I enjoy helping other people with their analyses. I seem to be better at helping people than doing sometimes. And I think it's just kind of come from that. It's something I definitely want to do more. Lots of people find it useful, which I find very rewarding. The presentation and everything is just because I think you need it to be clear, as clear as possible. Again, it's probably something I enjoy doing, maybe even more than it's worth. But you know, making the website's a lot of fun, and it is a lot of work, and some parts of it are not fun. Keeping the documentation up to date and making sure you're testing everything, and you take on a lot of responsibility when other people are using the software. But so far, it's all good. Um, there is no plan of commercializing GenoPred. Um, it's all open access, publicly available, which feels completely right because it uses loads of it's a pipeline using other software that people have developed and made publicly available, other brilliant software, which you know, my aim is just to broaden accessibility to it and make the workflows more reproducible.
Yeah.
So yeah, that's my standpoint.
Well, I'm sure everybody here will thank you for that. That's good news. Well, would you have any words of advice for anybody on the call who wants to develop their own software? I mean, how do you go about doing that?
So, and yeah, I mean, the other thing I would say is that I'm glad it seems professional because I very much have learned as I've gone. I've put a lot of time into it. It has been years now. So, but I mean, find other software you like and see how they work. And there's lots of incredible documentation online. I mean, don't go this way if you can't already do most of it, but ChatGPT is also great for certain parts like writing tests and documentation. Um, and yeah, I mean, yeah, exactly. Just, you know, it all kind of, I would say, started when I was using the software called Precise, and learning how that works, and then using other software like Fusion for TWAS, that's all written in R. And then you know, using the pipeline thing was something that a colleague introduced me to, um, which which again I just really enjoyed, and there's lots of documentation online of how to set these up. So yeah, I just put it down to people I've worked with and other publicly available software.
Yeah, great. Good. Thank you. That's great. We don't seem to have any other questions. So I guess if I mean, I think this is something that people will take away and use, and I think other people will watch this particular presentation offline too, as it's never the right time in several parts of the world at once to for actually everybody to come. So many people watch the talks offline too. So it's last chance for any questions before we wind up. I don't see any hands up, but there's a real spread here, Ollie, of people from across all career stages. And Andrea has just turned on her camera, so maybe she has a question. No, you don't. Sorry. Okay, sorry. Um, thank you very much, Ollie. That's been a brilliant talk. Apologies to Andrea for calling her out, but thank you very much for a really interesting talk and a great piece of software. So thanks again, and thanks to everybody who's able to come to this